Ivy153 coming at you again with another video about plants. I'm wearing this very fancy hat today because my hair doesn't look great, but I still wanted to introduce you to some very special guests that I brought with me today. If you've seen any of the pictures of my house on Instagram, you know that I have a lot of house plants. I love plants, so I thought I'd take this time to introduce you to some relevant members of my family. This first one I'd like to introduce is Ficus carica. This is a fig, and you can tell by the leaves that it has here. This is a really characteristic leaf shape for figs, and this is a fig that'll actually produce fruit. The variety of fruit that this little fig produces is called Violette de Bordeaux. It's very fancy. It hasn't produced fruit yet because it's still a little tiny baby, but I have multiple fruiting figs in my house. This is just one of them. The other special guest on our show today is this really cute succulent that I have. It's one of my favorites. It started out upright and then it kind of snaked over the side, and now it's like one of my favorite plants. It's got a little bit of purple on it, and this was a gift to me from SVC biology grad Annie Mills. Hi Annie, if you're out there watching this video, or Sarah, your sister. This is one of my favorite plants that you gave me, and this is a succulent. And you will see very soon why succulents and non-succulents are relevant, because we're going to talk about the different metabolic pathways that they use today. So, okay, bye little guy. Time to start talking about photorespiration. Okay, so we finished off our last video talking about a little bit of the nitty-gritty of photosynthesis. Let's talk a little bit about what happens when things might go wrong in photosynthesis. So let's talk about photorespiration. Photorespiration is when plants take up oxygen and give out carbon dioxide, so basically the opposite of photosynthesis. And it's a problem for plants when this happens because it wastes all that really good RUBP that we talked about before that is made during photosynthesis. Photorespiration happens because there's some quote-unquote flaws in the enzymatic activity of rubisco. There's a major problem with the actual molecule of rubisco. While it's able to attach to carbon dioxide molecules, it doesn't actually bind very well to carbon dioxide. In other words, you could say that rubisco has a low affinity for carbon dioxide. This means that when CO2 concentrations are low, there's also going to be low carbon fixation happening, which means photosynthesis isn't working at its optimum capacity. Another problem with rubisco is that in the presence of high oxygen concentrations, rubisco not only stops the carbon fixation reaction, but it actually starts to break down RUBP and starts forming carbon dioxide. So that's another problem, is you don't want it using oxygen. It's basically reversing all that hard work that was done during photosynthesis. So when does this happen? Why would this even happen in the first place? So when this normally occurs is in geographic areas that are extremely hot, and in areas where there's generally a lot of sunlight. Plant cells start to actively break down water to extract electrons for the light reaction. So their photosynthesis is like maxing out, and it needs all those electrons, and so it starts breaking down water for those light reactions. And if you're breaking water for energy, you're going to increase the amount of oxygen that's in the cell. And then if you increase that oxygen, then rubisco is going to start using that oxygen instead of fixing the carbon dioxide like it's supposed to. Additionally, the energy can initially be used by rubisco in the form of glucose, which means the concentrations of carbon dioxide inside the cell will begin to fall, increasing the ratio of oxygen to carbon dioxide in the cells. And as this occurs, rubisco will stop carbon fixation and it'll start doing photorespiration because of that change in concentration of CO2 and oxygen. And Although there's a lot of energy available to the cell, very little is going to be used in the formation of glucose. So, how does this translate into a problem for the plants? C3, okay. I talked about that. Okay. So, how do plants deal with that? How do C3 plants deal with that? So far what we've talked about is photosynthesis in C3 plants. C3 plants in hot, dry areas will have a difficult time fixing carbon, and changes in the cells will lead to increased photorespiration. In C3 plants that we've talked about so far, in just normal photosynthetic plants, Rubisco is the only tool that the plant has in its toolkit for fixing carbon. And so, because that's the only tool that it has, that's why you can sometimes get photorespiration. So what's actually happening when the sun is hot and the air is dry? The plants are going to respond to those environmental conditions by closing the stoma, which are the holes at the bottom of the leaves, which they use for gas exchange. We're going to see some pictures of those in just a second. And they'll close those holes to prevent water loss. They don't want to lose water, which is valuable to them when it's hot and dry. So when the stoma are closed, there will be an increase in the concentration of oxygen in the cell, which means Rubisco is going to start doing photorespiration, which makes photosynthesis less efficient because Rubisco is starting to eat up all that good stuff that photosynthesis was making. So what can the plants do to combat this? What they have to do is they need to change the means by which they fix carbon, and they need to find a way to continue to fix carbon in the presence of low CO2 and high O2 in the cells. And that's where evolution comes in to save the day. So there's two evolutionary novelties that have evolved to kind of fix this problem of photorespiration happening in dry and arid environments. The first solution is spatial, and the other is temporal. So let's talk first about the spatial solution, which is C4 photosynthesis. So in C4 plants, the process of carbon fixation and the other reactions in photosynthesis are separated into different cells. This process involves the addition of a second pathway by which the cells are able to attach the CO2 to an organic molecule that's going to help shuttle it to the Calvin cycle. So that's what you're seeing here is the spatial separation of attaching CO2 to an organic molecule, and that's happening here. So in C4 cells, the cells that are closest to the surface of the leaves, those are these mesophyll cells. They're going to use a 4-carbon molecule to attach to and fix the carbon dioxide, and then pass it to the cells further away from the surface to allow entrance into the Calvin cycle. Because the enzymes involved in fixing carbon are able to bind to carbon dioxide at a lower concentration, photorespiration doesn't happen. The most important molecule that I'd like you to know in this pathway, because it's also involved in CAM pathway, is malate or malic acid. That's this shuttling molecule that is attaching to the CO2 and shuttling it into these bundle sheath cells so that it can be used in the Calvin cycle. So the plants that do this, these are called C4 plants. They're able to maintain a high level of carbon fixation while keeping their stoma closed for a longer period of time and lowering their water loss. So it's a really cool strategy for dealing with arid dry environments with lots of sunlight. A lot of the plants that use C4 synthesis are actually very important agricultural plants. So corn and sugarcane are some really great examples of C4 plants that are very hardy because they have this C4 photosynthesis pathway. So that's C4 photosynthesis. That's the spatial solution to this photorespiration problem. The other one we're going to talk about is the CAM pathway. I just usually call it the CAM pathway because I can't really pronounce it. I'll try to. crassulacean acid metabolism. I hope I pronounced that right. You can just call it the CAM pathway. Don't worry about memorizing that name. CAM plants have developed a means to prevent the loss of efficiency through the process of having both C3 and C4 processes active in the cells, but they separate them in time. So they're doing things at different times of the day to help fix this photorespiration problem. Yeah, I said that. And they're able to separate these two processes into night and daytime cycles with the use of that storage molecule malate. So during the day, the stoma are closed, and the plant is cycling through the Calvin cycle. And it's breaking down that malate to get the carbon dioxide that it needs to fuel the Calvin cycle. And then at night, when the air is cooler, they're going to open up those stoma again to let moisture into the cell and breathe in that air. And that's when the cells will be able to open their stoma, allow this carbon dioxide to flow in, and then they're going to bind that carbon dioxide to malate so that it can then be used by the Calvin cycle during the day. So kind of an analogy to think about in these two different pathways is that you can kind of think about C4 plants as being commuter plants. So the carbon dioxide is taking a bus to its job every day. That bus is malate. So carbon dioxide is getting on the malate bus, and it's traveling from the mesophyll cells to their job in the Calvin cycle that's happening in the bundle sheath cells. So carbon dioxide hops on the bus right here, which is malate. That malate bus takes it from its home in the mesophyll cell to its day job in the bundle sheath cell where it performs the Calvin cycle, and that's how it transfers. So you can think of the C4 plants as using carbon dioxide as a commuter in C4 plants. And then the CAM plants, carbon dioxide works at home. So in these cells, CO2 is going to go to sleep in its little malate bed at the end of the day, and then and that's where it works. And then the Calvin cycle is its home office during the day. So at night, you've got CO2 attaching to malate and sleeping in its little malate bed, and then it wakes up during the day, and it goes to its home office in the Calvin cycle. Hopefully that analogy that I just made up didn't confuse you more and actually made you understand it better. Let me know on Schoology. Okay, so some examples of CAM plants are cacti and pineapples, and it's very likely that my little succulent friend that I showed you earlier is also a CAM pathway plant. And then my fig that I showed you earlier is a C3 plant. So here's a different illustration of the structure of the cells in C3 versus C4 plants. In the C4 plants, the mesophyll cells surround the bundle sheath cells so that they can bind that CO2 to malate before they shuttle it into their day job in the Calvin cycle inside the bundle sheath cells. So you've got these mesophyll cells that surround a bundle sheath cell. But in C3 plants, you don't have that kind of structure because you've got all the cells performing C3 photosynthesis. There's no spatial separation of these different chemical processes, chemical pathways. And then this is just another illustration of the difference between C3 plants and C4 plants. You've got the carbon dioxide coming in, jumping on the malate bus in the mesophyll cells, and then heading on its malate bus to its day job in the Calvin cycle inside the bundle sheath cells. In this one, you don't have that malate shuffle. The CO2 is just going straight into the Calvin cycle. And this is just another illustration of CAM plants and how they have the temporal separation. So at night, you've got that CO2 coming in to attach to its cozy little bed, which is malate, this organic acid. It's coming in here to bind, and then during the day, it's jumping out of its malate bed and going to its home office in the Calvin cycle and doing its job here. So I'll say that again. So the CO2 comes in, it binds to the malate where it's stored during the night, and then during the day, that CO2 is detached from the malate, and then it's used to function in the Calvin cycle. So the stoma are closed during the day and open at night when it's cooler and there's more moisture in the air. So if plants can just live better and operate at a higher capacity with the use of the C4 cycle, then why don't all plants use this in this process? The most basic answer to that question is that there's a lot of extra ATP involved in C4 and in CAM pathway plants in the process of fixation, which means that under some conditions there's still an advantage to not having to need all that ATP. And so there is still an advantage to C3 photosynthesis. So in highlight areas like the desert or in tropical regions, there's going to be enough extra light to supply the excess ATP needed in the fixation process. So this extra energy that you need to power C4 in the CAM pathway, it ain't a problem in these desert and tropical regions. But in areas like western Pennsylvania and the East Coast and temperate regions like the United States, we don't have as much sunlight availability, which means the plants won't be able to fix carbon as efficiently, and therefore they're going to suffer growth performance if there is C4 or a CAM plant. That's why you don't see a lot of cactuses growing in western Pennsylvania. There is actually one cactus that's native. There's an opuntia cactus. But other than that, you're not seeing a lot of cactuses growing around here because we don't have all that extra light available to power that CAM pathway in those plants. Most C4 and CAM pathways will actively function to keep the stoma closed most of the time, and this is a means to prevent water loss in warm temperatures. So in cooler temperatures, when there's not as much of a concern for water loss, they can keep their stoma open more, which means there's better gas exchange, and this means that just the addition of that extra step in the C4 and the CAM pathways will just make them less efficient than the C3 plants. So it ain't all bad to be a C3 plant, like my little fig that I showed you earlier. So water conservation in the case of both the C4, which keep their stoma closed most of the time, and the CAM pathways, which open the stoma at night, they're able to conserve and better use water resources available, which is why this is an adaptation that evolved in desert and arid regions, because it lets them trap that water and use it more efficiently when they don't have access, when water availability is very limited. So we'll talk about plant adaptations in the next few lectures, but you should be able to see that these C3, C4, and CAM plants each have specific types of environmental conditions that are going to favor their growth because they have highly adapted to those environmental conditions that they're found in. And so that's it for photorespiration. Our next lecture, we're going to move on to the next section and talk about plant structure and function. So just a mini lecture for you to cover. Until next time. Okay, see ya!